My research project is looking at gene expression's signature in this very common non-Hodgkin's lymphoma, which constitutes about 30% of all adult non-Hodgkin's lymphomas diagnosed in the West. Unfortunately, only just about half of patients can be cured, and those who are refractory upfront or die, or I should say relapse after complete remission, eventually do die. The diagnosis of diffuse large B-cell lymphoma hasn't changed very much over the years, and the prognostication, despite being adjusted for age and more recently with rituximab, aren't very good surrogates or precisely define the underlying biological heterogeneity of this condition that looks the same under the microscope. And because of next generation sequencing, the relative ease of arraying fresh tissue, there's been a movement to try and reclassify diffuse large B-cell lymphoma, particularly over the last 2 or so decades. The most, I guess, validated and clinically applied classification model is based on the putative origin of the cell that the lymphoma arose from. However, there are other classifications that have been published and have shown some promising results based on the type of metabolism, such as oxidative phosphorylation that the lymphoma cell employs, particular signalling pathways, particularly B-cell receptor and the downstream cascades, the underlying inflammatory milieu, be it T cells, the presence of angiogenesis and the presence of fibrotic cells. However, the most commonly known is based on B-cell ontology, which is the cell of origin classification. And since this very highly cited publication in Nature over 15 years ago, there have been many groups that have replicated this, but mainly in retrospective and relatively small studies. And it's generally accepted now that diffuse large B-cell lymphoma, not otherwise specified, looking exactly the same under the microscope, can be classified in terms of germinal centre-like diffuse large B-cell lymphoma, which express the same coordinated set of genes that a normal B-cell would express during its transit through the germinal centre. Or it could be an activated B-cell-like gene signature, which is a set of genes that are expressed when in vitro stimulation with a mitogen, and the lymphoma cell expresses those type of genes. And the primary mediastinal B-cell lymphoma has been shown to be a different type of lymphoma, so I've put that in brackets. This cartoon is a recent publication that summarises the very well-known biological pathways that are preferentially exploited by the different types of lymphomas, which I'll call GCB and ABC from now on. And some of these are now molecularly targeted, with drugs being in the pipeline. However, is cell of origin important? Because definitely locally, I think, having trained through various hospitals, I don't think it is applied, particularly clinically. I've put up the survival curves from a very well-cited paper published in 2008 by the Leukemia Lymphoma Molecular Profiling Project. But actually, since the cell of origin was discussed in the late 90s, there's been a recognition that people with the ABC type of diffuse large B-cell lymphoma do much worse, and this has been replicated in the R-CHOP era. And as a result, there is currently a prospectively trial undergoing recruitment in the United States. It is sponsored by industry, by Celgene, where the recognised form of lymphoma that does poorly, ABC, is going to be treated upfront with lenalidomide, and this will be the first prospective multi-centre trial looking at the importance of ABC versus GCB classification at diagnosis. Unfortunately, it has been hard to translate cell of origin into clinical practice because most of the technologies, be it massively parallel sequencing or microarrays, have required fresh tissue. And the cost, the technology, as well as the turnaround time have been prohibitive for implementation into clinical practice. So immunohistochemistry have been used as a surrogate, but it's an imperfect surrogate, but it is accessible. There are various algorithms that have already been mentioned. Tally currently is the most well-validated algorithm, but the Hans and the Choi are also used. However, they all have inherent pre-analytical and analytical issues, and the positive predictive value is well less than 1 compared to standard gold standard, which is Affymetrix. So I am privileged to be doing my postgraduate training at a centre that has acquired a nanostring encounter platform. And it's a relatively new technology. And it does not require amplification or enzymatic modification to extract nucleic acid, which has been the main barrier for profiling genes or genotyping in both solid organ and hematological malignancies. It uses probes, which I have here is probe A and probe B, that bind to DNA or RNA sequence of interest, and it's captured by a tag, which is called here, and onto a slide by a biotin streptavidin very strong bonding, and then it is counted by a camera essentially in the machine through a reporter probe. And so it uses hybridization technology to capture the nucleic acid sequence of interest, and then it digitally counts and produces a count of the genes that one is interested in. So far, this technology has been used mostly in the research setting, as is more described for solid organ malignancies. However, in the lymphomas that we're interested in, there was a paper that was published in early 2014 by the authors that constitute the LLMPP, and they were able to accurately define cell of origin, and the comparator was Affymetrix as well as immunohistochemistry using just 15 genes. It says 20 genes there, but 5 of them are actually housekeepers. And so what my project aims to do is to translate this panel that uses 15 genes, 5 housekeepers, to assign cell of origin to lymphoma samples, and to explore other molecular features of diffuse large B-cell lymphoma. The genes that we've been interested in all along are c-Myc, BCL2 and BCL6, because they are very well known and have independent prognostic significance. But other gene signatures that I've become interested in through my literature review have been PIM kinases, hedgehog signalling, and genes that control metabolism, hypoxia, cell cycling, and these set of genes Dr. Stevenson actually has a personal interest in, and we're finding significant results as well. So what we've done so far is, through some of the background work that Louise as well as David, one of our previous registrars, have done, is identify samples of patients that have been treated with R-CHOP or R-CHOP-like chemotherapy and that have clinically annotated details. We've designed a panel that have incorporated the Scott paper, the Lymph2CX, through an academic material transfer agreement, as well as 28 other genes, so that we have 48 genes in total. And the 48 genes is mainly limited by funding issues. This panel can test up to 900 genes in one go, but we don't have the funding for that. And we have been developing a local protocol for genotyping tissue that is archived, have been fixed in formalin, and have been embedded in paraffin. And we've been using samples that vary in size, age, and the quality of the nucleic acids. And only about a fortnight ago, after all the regulatory and other logistical issues, we performed a pilot run. Apart from a normal lymph node control, we used 11 lymphoma patient samples. 9 were core biopsies, because that seems to be how most people are diagnosed at North Shore Hospital, thanks to radiology mainly, and only 2 were excisional biopsies. Mentioning the core, a lot of them were just one single core. There were 2 that had 3 and 2 that had 2 cores, and the smallest one had a surface area of only 5 millimeters squared. The specimen aged had a median of around just 5 years, ranging from 32 to 149 months. The RNA quantity was fair, with a median of 56 nanogram per microliter, which usually is okay in biological studies. And the RNA quality was uniformly poor. We defined this by 2 different methods, using the nanos drop, which uses spectrophotometry. The 260, 280 shows that it's very impure, that 1.91. We aim for something over 2 if it's something like PCR. And the 260, 230 shows that it's very contaminated. And it's contaminated because it was embedded in paraffin. And again, we aim for over 2 and the median is 0.8. And then we also borrowed a chip from a lab downstairs and we ran this through Agilent that uses flow cytometry and electrophoresis, and the RNA integrity index was very low, 10 being optimal, 5 being so poor that most people wouldn't do PCR on. So in terms of the results, answering our protocol aim of can we extract RNA and genotype, and also the study aim, the results were quite encouraging. We were able to profile the gene expression of all 11 samples plus to 1 control from tiny biopsies, from impure RNA that came from archival tissues, and the controls, the housekeeping genes, all worked and there were no QC flags. In terms of our study aims, we could assign cell of origin using the leukemia lymphoma molecular profiling project algorithm, which they have kindly lent us for academic use, in about 3/4 of the patient. About 1/4 were unclassifiable. And in terms of other gene expression signatures that we're interested in, the 28 other genes, we were able to observe some patterns, but of course the numbers are very small. Looking at what we are trying to do, this is very uninterpretable at the moment. These are the 11 patient samples, and this is the panel of the 48 minus 5 housekeeping genes, so 43 genes that we're doing. And we don't plan to analyse this in depth at the moment, just because the N is equal to 11. But I guess it's a quality control measure, looking at just the SCOT gene, or the genes used in SCOT paper, in the blood paper. The first lots of samples circled in yellow was defined to be germinal center B cell. The middle 2 in orange was activated B cell, and the last 3 were unclassifiable. And on our heat map, it was very pleasing that this conformed to what was predicted on the algorithm that the NCI had allowed us to use. The other 28 genes that we're interested in also seem to stratify the lymphoma samples into 2 big groups, but we haven't had time yet to really compare them to the previous dendrogram. So I won't go back to it. So far we can say that genotyping works from these very small biopsies. I thought I'd briefly show in some of the analyses I'm trying to do, but with a very small pilot sample, these 6 genes seem to be differentially expressed between ABC versus GCP with a statistically significant p-value using a 2-tailed significance test of 5%, with a relatively conservative false discovery rate of less than 10% as well. And I guess I've circled BCL2 and P10 here because those 2 pathways are very well known in B cell biology, and also there are molecules now that are trying to inhibit BCL2 as well as mTOR in the PI3K, AKT, mTOR pathway that P10 is a natural antagonist in. I'm just going to focus on BCL2, and one of the aims that we are going to try and do is to correlate cell of origin expression, ABC versus GCB, and those that are unclassifiable, with standard diagnostic immunohistochemistry, in which case all our patients showed IHC expression because IHC is a qualitative diagnostic. And in terms of whether BCL2 is rearranged molecularly on FISH, the percentages probably don't mean very much, but once we go through about the 200 or so samples, perhaps we'll be powered enough to compare genotyping with immunohistochemistry as well as FISH. Next, and I've highlighted this in red because I really shouldn't have done it because the sample size is so small, but I couldn't help myself. Looking at those who have died in the pilot sample, there were only 3 people, but I decided not to really look into it because 2 of them actually died in complete remission from non-Hodgkin's lymphoma. and so the numbers were too small to really correlate. In terms of those who relapsed, again, it's very hard to delineate whether the cell of origin really means anything. But looking at gene signatures that were differentially expressed between those who relapse and those who don't, there were 3 genes in the pilot that seems to show differences with significant p-value. So I'll be interested in seeing if this pans out in a bigger sample size. Louise had already mentioned MYC being a molecule or gene or protein of interest in our institution, and looking at the gene expression of MYC compared to IHC as well as fish is very heterogeneous, and so and as well as what Taz has mentioned already, we need to define what MYC's role in terms of gene and protein expression a bit more, and hopefully with a bigger study we can answer some of those biological questions. So, so far I don't have much results to present, but I was happy that our pilots showed that we could profile genes from small samples. There were a few issues that we have perhaps identified. The cell of origin assignment is based on an algorithm that is a web-based tool that we have been allowed to use. We don't know how they calculate it. It's based on a Bayesian theorem and with a lot of prior assumption. Since the publication in Nature, that group has been using the same algorithm ever since, and so from a translational point of view, perhaps it is okay that we use it without really knowing how they calculate it or the maths behind it. And another issue is of our 3 cases, they were unclassifiable, which is about 25%. We need to work out whether it's because of technical issues, maybe we're not doing the runs well enough, or is it because of underlying biological issues. And I subsequently found out it was because one of the samples that I did was actually a diffuse large B-cell lymphoma coming out of a colon, so perhaps I can't use the same panel of genes to diagnose or to look at gene expression in non-lymphomatous tissue. So where I'd like to go further is to validate this panel of genes in the entire cohort. We are limited by, I guess, finances. We're doing about 190 or so patient samples to generate some hypotheses from the other gene signatures and work with cell lines, and also perhaps implement this prospectively through further clinical studies and maybe if there are biological agents that we can use. Lastly, I'd just like to thank quite a few people, particularly Will here, who's my supervisor, and Sydney Vital, who provided funding for about half the consumables of the project. Thank you.